Thanks, Chris. And thank you all for being here. A lot of great talks, really enjoyed it today. So I'm going to be talking a little bit about our monitoring methods and efforts on Great Bay on horseshoe crabs, and have a little bit of new information and new methods today. So why monitor horseshoe crabs? They're keystone foundational species in a number of different estuaries all along the East Coast. They're bioturbators, as you can see in the top right. Those pits are late summer, made every one of them made by horseshoe crabs, so they turn over a lot of mud. Their eggs are also really important key forage for a lot of species, including long distance, critically endangered species like red knots. And they're part of two fisheries, both bait and blood. Their blood, as you can see in the bottom right-hand corner, is used as the most sensitive assay for gram-negative bacteria. And they are used for bait for both eel and conch. And why monitor? Because we are seeing some population declines in some areas. Most states do survey. New Hampshire doesn't have an official survey. And interestingly, there's no or little or no harvesting in Great Bay. So it seems like a great control site to look at other effects such as climate change and loss of spawning beaches. And we have seen that in Great Bay. So what are some possible problems with current monitoring? Well, current monitoring up and down the seaboard involves beaches, and beaches really make up a very small percentage of the edge shoreline. Other habitats such as marshes, a very much higher percentage. And we have observed horseshoe crabs here, as you can see in this bottom right, that is high marsh and low marsh. And we see a lot of crabs here, but are they just visiting? Are they lost? Or are they actually spawning? So the goal is to develop some new monitoring protocols and include marsh sites. And I'll get this a little bit later, use sediment coring to potentially, as a better measure, measure and assess egg density. So here's the requisite map of Great Bay and our monitoring sites over 8 years or almost 15 years throughout Great Bay and Little Bay. And you're familiar with those areas. We've added a couple of new sites with funding from U.S. Fish and Wildlife Service grant. That includes marshes. A lot of people out here have seen horseshoe crabs in marshes, but including myself and my colleague Wynn Watson and others. But are they just visiting, or are they using these areas? So we picked out a couple of areas, Great Bay Farm and Pickering Brook, and monitored these areas. So how do we assess density of beach visits? We take or lay out 100-meter transects in general and count them during high tide and count the animals, males and females, and tot those up. And here are some data from our 6 beach sites. And you can see over about a 5-year period, there's some variability, but it's fairly stable among these several sites. And then again, not surprisingly, because a number of people, including us, have seen these in other areas, we do see them in the marshy sites as well, including Pickering and Great Bay Farms. And these numbers are pretty similar to what you see in the beaches. And I mentioned that this is part of the Fish and Wildlife Service grant. And we also looked at these, had collaborators down in South Carolina and in Connecticut, and in those areas as well. We see that there are females both in the beach and in the marsh. You can see fairly high densities in the marsh, very similar averages between the two sites, but larger variance pretty clearly in the marsh. So they're there, but are they spawning? That's really important. So we decided to assess spawning, and to do that, we again took laid out 100-meter transects, took sediment cores every meter, and then took them back to the lab, washed them, and counted the eggs. And the results are pretty surprising. Yes, they do use the marsh. This is a paper in Press, and maybe even slightly higher densities in these marshy areas compared to the beach. If you just do the math, it's almost 100,000, that top point up there, about 100,000 eggs per square meter, which is really remarkable. So they are using the marshes. Similar egg densities, maybe a little higher even, but really importantly, do they survive and develop? Because we all know that those areas, sediments are fairly anoxic, at least we think, at least some of the sediments, right? So do they survive and develop? So last year we started marking nests, and then coming back 1 or 3 weeks later and digging up those nests to look at development. We marked about 50 nests per site and went back, dug the eggs up and staged the animals. And we have from A to G increased levels of development. And when we assessed the eggs, these data now are just from Great Bay. This is a paper in prep. We can see similar development in both the marsh and the beaches. Not much going on yet. You can see a little bit in stage B, very similar in both ways, but very importantly, when we come back 3.5 weeks later, we see development in those eggs as well. So not only are they using these marshy sites, but the eggs are developing, and the animals seem to be doing pretty well. Very similar numbers across these different developmental stages, including importantly those at one criterion black. That's where they have essentially died. Very similar amounts of eggs seem to die between the two different groups. So why spawn in marshes? Well, in Great Bay at least, is very limited beaches. Less than 1% of the edge is probably beach. And one of our major beaches that we have done counts on for many years has really disappeared on the southern part of southwestern part of Adams Point. So they seem to be disappearing. If you know of any that are uphearing, I'd really like to know about that. So we were wondering whether a whole host of abiotic factors may be involved, and one of them is temperature, of course. And so we planted hobos in the different sites and looked at them, and lo and behold, at least in Great Bay, it seems that temperature does seem to be a little bit warmer in the marsh sediments than in the beaches, perhaps allowing for quicker development, but I'll remind you that we didn't really necessarily see quicker development in our data. Right. So summary, horseshoe crabs do spawn in marshes, a whole host of questions. How prevalent is this? We've looked at a couple of marsh sites and found eggs. We've also looked at a lot of marsh sites and not found eggs. So one of the things we'd really like to tap into all the expertise out here is, where do you see horseshoe crabs? Where else might we go and look for these? And we're especially interested in the sediments and the sedimentology. So I've talked to a few of you today, really a great session today, to try to learn a little bit more about that. What factors are involved with how they site select on these marshy areas? Do subpopulations have habitat preferences? Do we have beach breeders and we have marsh breeders? Where might additional areas be? And might this help to decrease egg predation? Lots of questions. Marsh eggs do develop about the same rate as beach eggs. We don't know if they hatch though. We haven't done that level yet. I'd like to go out in next couple of years and do some netting and plankton netting and look to see if they are in there. Anecdotally, we do see trilobite larvae in the water column at some of these areas, so we think that that's going to turn out to be true. And lastly, cores might be a better way, or at least an adjunct way, to monitor horseshoe crab populations. It is a bit harder to process each sample, but you can do it anytime. You don't have to be there at high tide during the day to count crabs. And the data seem to be as good, very short data set here, and arguably better because we do find crabs at a number of marshy sites, but we don't necessarily find eggs there. So I'll be happy to take your questions.
Ed, Tessa Corsetti, Great Bay. What's the process when you guys bring the eggs back to the lab? Are you able to return them after you count them, or?
We actually do, yeah.
Awesome.
It's pretty easy to wash them, count them, and then return them. They keep very well in the lab.
Pretty hardy.
They're very hardy, and we actually use a subset for behavioral work over the summer. So they do very well on the transport, no problem at all there.
Okay. Chris, you would add Greg Moore, you had mentioned you wanted know about sites that you're seeing them at. And the curious thing, when we put out our pilot study for thin layer placement of sediment in the playpens that Marjorie was showing, not in her experiment, but the previous one, we repeatedly had horseshoe crab burrowing going on in these little sandboxes. And we just, didn't think too much about it until now, as it were.
Interesting.
And you just, you could see the outline and clearly the body, carrots like shaped right in there, and it was common in our location. So maybe they were just visiting, but they found some cool places to do their thing.
Right, so the question is, were they spawning or were they feeding? Because they throw a lot of mud in both cases, yeah. Thanks.
I'll keep you posted on.
Thank you, yes, I'd really appreciate, my email is listed, so I'd really like to hear where those other sites might be.
Chris, Steve Jones, UNH. By the way, I have that map, that Elvis map too. So the site selection, is it, to what extent is it dependent on the texture of the settlement?
We think a great deal, and that's why I want to try to quantify what sites they like and what will they be willing, where can they really lay eggs? What we've seen so far, Steve, is that there's plenty of area in low marsh, especially along tidal creeks. So Great Bay Farms and Pickering Creek are both tidal creeks. And there's an area, I'm Frenchman, so I'll use my arms here, about this wide, right, of low marsh where they will lay their eggs. Now, they could be going lower, we can't see. You can't see, the turbidity of that water is very high, so you can't really tell, right? But nothing in the high marsh. And when we started taking cores, I mean, to get a core in a high marsh, you gotta get up there with all 200 pounds of you and drive it down, and really it takes a lot of work to do that, whereas in low marsh, pretty easy digging. Another question is why is the environment okay for them to develop? Oxygen needs of developing critters are pretty low, but still they need some. And we know, quote unquote, at least I do as a biologist, other people might disagree, that fairly anoxic conditions underneath that mud.
One last question.
Animal Forest, UNH. When you mentioned at the beginning that we're seeing declines kind of along the East Coast, do you expect a fishery to develop up here for blood and bait in the coming years?
So no, probably not. I believe the blood industry is going to go away. We have a very good substitute right now. It's just the pharmaceutical companies are resistant to enacting that, industry being, very conservative when it comes to these things. In terms of bait, our numbers are pretty good in Great Bay, but they're nowhere near what they are in other areas, especially south of Cape Cod, and that's where the industries are generally located. So I don't, putting on my, or looking into my crystal ball, I don't see that happening. Thanks for the question. All right, thanks very much.